In this video, we will understand the structure of DNA, the molecule that carries genetic information in all living organisms. DNA is responsible for heredity, variation, and the transmission of traits from one generation to the next. DNA stands for deoxyribonucleic acid, a long molecule that stores the instructions for building every living thing. In eukaryotic cells, these instruction manuals are kept in the nucleus, cell's command center. In addition, small amounts of DNA are also present in mitochondria and chloroplasts. Now here's the most fascinating part. DNA is an extremely long molecule. If stretched out, the DNA from just one cell would be about 2 meters long. Yet, it fits inside a nucleus that is smaller than a dot on paper. How is that possible? This happens because DNA is tightly packed. First, the DNA wraps around special proteins called histones, forming bead-like structures. These beads coil again and again, becoming thicker and denser, until they form chromosomes. Each DNA nucleotide consists of 3 components: a phosphate group, a deoxyribose sugar, and a nitrogenous base. These components together form the basic structural unit of DNA. DNA contains 4 nitrogenous bases: adenine, guanine, cytosine, and thymine. These bases store the genetic information in the form of a specific sequence. The sugar-phosphate backbones stay on the outside, while the paired bases form the steps inside, just like a twisted ladder. This pairing is maintained by hydrogen bonds, which stabilize the DNA structure. The nitrogenous bases are classified into purines and pyrimidines. Purines, which include adenine and guanine, have a double ring structure. Pyrimidines, which include cytosine and thymine, have a single ring structure. Now notice the order of bases along the DNA strand. This sequence, TGAACTGGT, is not random. It is extremely specific. Each gene is defined by a unique sequence of nitrogenous bases. This exact sequence decides which protein is made, how it folds, and how it functions, and that ultimately determines the traits, health, and fate of an organism. Rosalind Franklin produced the famous photograph 51A X-ray diffraction images that revealed DNA's helical pattern, a vital clue for solving the structure. Using Franklin's data, James Watson and Francis Crick, along with contributions from Maurice Wilkins, proposed the correct double helix model of DNA in 1953. DNA can exist in different structural forms depending on environmental conditions. These include A, B, C, and Z DNA, each with distinct structural characteristics. aDNA is a right-handed helix with 11 base pairs per turn and a pitch of 28.2 angstroms. It is usually formed under dehydrated conditions. bDNA is the most common and biologically active form of DNA. It is right-handed, has 10 base pairs per turn, and a pitch of 34 angstroms. cDNA is a right-handed helix with 9.3 base pairs per turn and a pitch of 31 angstroms. It is less common and forms under specific ionic conditions. zDNA is a left-handed helical form with 12 base pairs per turn and a pitch of 43 angstroms. It has a zigzag backbone. If this peek into DNA sparked your curiosity, hit like and subscribe. Next time we'll animate replication and see how cells copy this incredible molecule.